Hey guys, welcome back to my channel. This is Dr. Rajeshwar from YR PharmaTube. In the previous video, we have learnt about 11 different types of oxidation reactions catalyzed by cytochrome P450 enzyme. If you did not watch the videos, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and medicinal chemistry, click on the links given in the description box below this video. In this lecture, we shall try to understand the oxidation reactions not catalyzed by cytochrome P450 enzyme. At the end of this video, you should be able to understand the different types of phase 1 drug metabolic oxidation reactions which are not catalyzed by the cytochrome P450 enzyme. But these are specifically catalyzed by alcohol dehydrogenase, aldehyde dehydrogenase, xanthine oxidase, amine oxidases, aromatase, and alkyl hydrazine oxidase enzymes. Unlike to the oxidations discussed in the previous videos, some oxidation reactions are catalyzed by hepatic enzymes localized outside the microsomal system, which means that there are certain drugs that can be oxidized by a number of enzymes in the human body not related to cytochrome P450. These enzymes include alcohol dehydrogenase, aldehyde dehydrogenase, xanthine oxidase, amine oxidases, aromatase, and alkyl hydrazine oxidase enzymes. These enzymes tend to be more specific, oxidizing xenobiotics related to the normal substrate for the enzyme. Most of these enzymes are primarily involved in endogenous compound metabolism. Number 1. Alcohol dehydrogenase. Alcohol dehydrogenases are abbreviated as ADH. They are a group of dehydrogenase enzymes that occur in many organisms and catalyze the oxidation of many alcohols to the corresponding aldehydes. Thus, they serve to break down alcohols that are otherwise toxic, and they also participate in the generation of useful aldehyde, ketone, or alcohol groups during the biosynthesis of various metabolites. For example, an alcohol, ethanol, can be oxidized to its corresponding aldehyde, that is acetaldehyde. This oxidation involves the loss of two hydrogen atoms. This reaction is catalyzed by the enzyme alcohol dehydrogenase and is dependent on NAD+ as a cofactor. In the mechanism of oxidation, a hydride ion is transferred to NAD+ to form NADH. Alcohol dehydrogenase is one of the major enzymes involved in ethanol metabolism and may have a protective effect against alcoholism. The enzyme alcohol dehydrogenase is localized in the soluble fraction of liver, kidney, and lung cells. Unlike the microsomal ethanol oxidizing system mentioned above, this enzyme uses zinc-containing NAD+ as a cofactor, and it is a true dehydrogenase enzyme. As discussed earlier, the enzyme ADH is used in the oxidation of alcohols to aldehydes or ketones. Oxidation of primary alcohols lead to aldehydes, and oxidation of secondary alcohols lead to ketones. Although alcohol dehydrogenase is the major enzyme involved in the oxidation of ethanol and most other low molecular mass alcohols, cytochrome P450, especially 2E1, can also oxidize ethanol, and this enzyme is induced in alcohols. Although comprehensive studies have not been published, it appears that cytochrome P450 are often the major enzymes involved in the oxidation of higher molecular mass alcohols. The native animals, the cytochrome P450 mediated oxidation of ethanol is thought to be of minor importance. But following induction of ethanol, the microsomal oxidation of ethanol increases dramatically and may account for 80% of ethanol clearance in certain cases. In non-induced situations, alcohol dehydrogenase is the major metabolizer of ethanol. In an example, an anti-anxiety drug hydroxyzine is broken into its active metabolite citrizine by alcohol dehydrogenase enzyme. Other drugs with alcohol groups may be metabolized in a similar way as long as steric hindrance does not prevent the alcohol from reaching the active site. It is to note that alcohol dehydrogenase activity varies between men and women, between young and old, and among populations from different areas of the world. For example, young women are unable to process alcohol at the same rate as young men because they do not express the alcohol dehydrogenase as highly, although the inverse is true among the middle aged. The level of activity may not be dependent only on level of expression but also on allelic diversity among the population. Number 2, aldehyde dehydrogenase. Aldehyde dehydrogenases are a group of enzymes that catalyze the oxidation of aldehydes. They convert aldehydes to carboxylic acids. In the reaction, the oxygen comes from a water molecule. Aldehyde dehydrogenases found in systolic are belonging to class 1, those belonging to mitochondrial are class 2, and those expressed in tumors, stomach, and cornea are belonging to class 3. These are all responsible for the oxidation of aldehydes to carboxylic acids. In all three classes, aldehyde dehydrogenase 1 and aldehyde dehydrogenase 2 are the most important enzymes for aldehyde oxidation, and both are tetrameric enzymes composed of 54 kilodaltons subunits. All these enzymes are dependent on either NAD+ and NADP+, which functions as a cofactor. Till date, 19 aldehyde dehydrogenase genes have been identified within the human genome. These genes participate in a wide variety of biological processes, including the detoxification of exogenously and endogenously generated aldehydes. In this NADP plus dependent reaction, the aldehyde enters the active site through a channel extending from the surface of the enzyme. The active site contains a Rossmann fold, and interactions between the cofactor and the fold allow the action of the active site. A sulfur atom from a cysteine in the active site makes a nucleophilic attack on the carbonyl carbon of the aldehyde. The hydrogen is eliminated as a hydride and attacks NADP plus to make NADPH. The enzyme's active site then goes through an isomorphic change whereby the NADPH is moved, creating room for a water molecule to access the substrate. The water is primed by a glutamate in the active site, and the water makes a nucleophilic attack on the carbonyl carbon, kicking off or eliminating the sulfur as a leaving group. Aldehyde dehydrogenases can also reduce quinones to hydroquinones. The products are considered to be potentially toxic. Aldehyde dehydrogenase levels are elevated in tumors. Aldehyde dehydrogenase 3 is cytosolic tumor specific and is involved in activation of cyclophosphamide, mitomycins, and anthracyclines to their respective toxic or active metabolites. Number 3. Xanthine oxidase. Xanthine oxidase is a form of xanthine oxidoreductase, a type of enzyme that generates reactive oxygen species. These enzymes catalyze the oxidation of hypoxanthine to xanthine and can further catalyze the oxidation of xanthine to uric acid. These enzymes play an important role in the metabolism of purines and purine analogues and are xanthine containing drugs such as caffeine, theophylline, and theobromine in some species, including humans. For example, in the form of the N9H tautomer, it undergoes xanthine oxidase catalyzed oxidation with high affinity for different regions. At C6 yielding hypoxanthine, then at C2 generating xanthine, and finally at C8 with the corresponding formation of uric acid that is excreted in the urine. Hypoxanthine contains one oxygen or oxo group at C6. Xanthine contains two oxygens or oxo groups, one at C2 and the another at C6. And in uric acid, three oxygens or oxo groups at C2, C6 and C8 positions. Xanthine oxidase can also act on certain other purines, pterins, and aldehydes. For example, xanthine oxidase efficiently converts 1-methylxanthine, a metabolite of caffeine, to 1-methyl uric acid but has little activity on 3-methylxanthine. Another reaction catalyzed by xanthine oxidase is the decomposition of S-nitrosothiols, a class of reactive nitrogen species, to nitric oxide, which reacts with a superoxide anion to form peroxynitrite under aerobic conditions. Xanthine oxidase has also been found to produce the strong 1-electron oxidant carbonate radical anion from oxidation with acetaldehyde in the presence of catalase and bicarbonate. It was suggested that the carbonate radical was likely to be produced in one of the enzyme's redox centers with a peroxy monocarbonate intermediate. Here is the diagram highlighting the pathways catalyzed by the xanthine oxidase enzyme. Number 4. Amine oxidases. An amine oxidase is an enzyme that catalyzes the oxidative cleavage of alkyl amines into aldehydes and ammonia. Amine oxidase enzymes can be subdivided into monoamine oxidases, diamine oxidases, and the flavoprotein N-oxidases and N-hydroxylases. Monoamine oxidases are responsible for the metabolism of endogenous catecholamines, for example epinephrine, norepinephrine, serotonin, etc. Diamine oxidases deaminates endogenous diamines, for example histamine, and the flavoprotein N-oxidases and N-hydroxylases have already been discussed in the previous videos, that is, the oxidations involving carbon-nitrogen bond. Monoamine oxidases are a family of enzymes that catalyze the oxidation of monoamines, employing oxygen to remove their amine group. They are found bound to the outer membrane of mitochondria in most cell types of the human body. This enzyme system brings about the dehydrogenation of an amine to an imine, which is then hydrolyzed to a carbonyl group. The monoamino oxidases belong to the protein family of flavin-containing amine oxidoreductases, which play an important role in the metabolism of the neurotransmitters such as dopamine and many of the drugs which interact with them. In humans, there are two types of monoamino oxidases, namely monoamino oxidase A and monoamino oxidase B. Both are found in neurons and astroglia. Outside the central nervous system, monoamino oxidase A is also found in the liver, pulmonary vascular endothelium, gastrointestinal tract, and placenta. Monoamine oxidase B is almost found in blood platelets. Monoamine oxidase enzyme A is particularly important in the catabolism of monoamines ingested in food. Both monoamino oxidases are also vital to the inactivation of monoamine neurotransmitters, for which they play different specificities. Epinephrine, norepinephrine, and serotonin are mainly broken down by monoamine oxidase enzyme A. Phenethylamine and benzylamine are mainly broken down by monoamine oxidase B. Both forms metabolize dopamine, tyramine, and tryptamine. However, some evidence suggests that monoamine oxidase B may not be responsible for a significant amount of dopamine degradation. Specific reactions catalyzed by monoamino oxidase enzyme include epinephrine 2, 3,4-dihydroxy-mandelaldehyde, norepinephrine to 3,4-dihydroxymandelaldehyde, serotonin to 5-hydroxyindole acetaldehyde, 5-hydroxytryptamine to 5-methoxyindole acetaldehyde, tryptamine to indole acetaldehyde, dopamine to 3,4-dihydroxyphenyl acetaldehyde, phenylethylamine to phenyl acetaldehyde, and benzylamine to benzaldehyde, and so on. Diamine oxidases are known as amine oxidases or copper-containing amine oxidases, which are formerly called histaminases. It is an enzyme involved in the metabolism, oxidation, and inactivation of histamine and other polyamines such as putrescine or spermidine. The enzyme belongs to the amine oxidase copper-containing family of amine oxidase systems. For example, diamino oxidase metabolizes histamine into imidazole 4-acetaldehyde. Imidazole 4-acetaldehyde is then further oxidized by a NAD-dependent aldehyde dehydrogenase enzyme, leading to the formation of imidazoline 4-acetic acid. Diamine oxidase is primarily involved with endogenous metabolism and is of little relevance here, whereas the N-oxidases are of importance in the metabolism of drugs, for example, imipramine. These enzymes are found in liver microsomes. They appear to require NADPH and molecular oxygen but are not mixed function oxidases. They are flavoproteins. Number 5. Aromatase. Aromatase, also called estrogen synthetase or estrogen synthase. It is an enzyme responsible for a key step in the biosynthesis of estrogens. It is cytochrome P19A1, a member of the cytochrome P450 superfamily, which are monooxygenases that catalyze many reactions involved in steroidogenesis. In particular, aromatase is responsible for the aromatization of androgens into estrogens. The enzyme aromatase can be found in many tissues including gonads, brain, adipose tissue, placenta, blood vessels, skin and bone, as well as tissue of endometriosis, uterine fibroids, breast cancer, and endometrial cancer. It is an important factor in sexual development. Aromatase is localized in the endoplasmic reticulum, where it is regulated by tissue-specific promoters that are in turn controlled by hormones, cytokinins, and other factors. It catalyzes the last steps of estrogen biosynthesis from androgens, especially it transforms androstenedione to estrone and testosterone to estradiol. These steps include 3 successive hydroxylations of the 19-methyl group of androgens, followed by simultaneous elimination of the methyl group of formate and aromatization of the A ring. In addition to the above, xenobiotics containing a cyclohexane carboxylic acid group can also be converted to the corresponding benzoic acid by a liver and kidney mitochondrial enzyme. The enzyme requires the coenzyme A, derivative of the acid, as a substrate and oxygen and FAD as cofactors. And number 6, alkyl hydrazine oxidase. Drugs containing alkyl hydrazine group can undergo oxidation in the presence of alkyl hydrazine oxidase enzyme. For example, carbidopa can be converted to 3,3,4-dihydroxyphenyl 2-methylpropanoic acid by oxidation of the nitrogen function and subsequent rearrangement and decomposition of the intermediate compound. The exact mechanism is not known. This is the list of references followed for the lecture. That's all in this video. The oxidation reactions not catalyzed by the cytochrome P450 enzyme. In the next lesson, we will discuss about the reduction reactions of drug metabolism. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.